In-class problem, discovery of drug side effect, Gatifloxacin, tutorial 25. For those who wish to get deeper insight into cohort building and its use in solving analytic problems in accountable healthcare, please read Riddles in Accountable Healthcare by Eran Bellin, available as paperback from Amazon in Kindle or in paperback. The objectives of this exercise: learn to build medication prescription sets with either Find or Explore. Learn how and why to use a subgroup. Learn how to use blackout period in Study Designer time to outcome method and why. Realize that you can recapitulate studies in the medical literature using clinical looking glass with local data. And this is the actual study you're going to recapitulate. Outpatient Gatifloxacin therapy and dysglycemia in older adults, the New England Journal of Medicine. The cohort criteria. The case group is to be made up of outpatients prescribed Gatifloxacin. The control group, made up of outpatients prescribed imacrolate. Both groups are going to be made up of patients older than age 65. We purposely choose older patients because they're more susceptible to the side effects. They have less homeostatic control, and we will see a side effect if it appears. The time period we're going to look at is January 1st, 2004 to March 6, 2006, inclusive of 1104, that's why the square bracket, and excluding 3606 on the right. The outcome criteria: we're going to have a glucose whose lab test type is known as a glucose combo, FSG plus glucose. You'll find it in the system that way, that is both a fingerstick glucose or a glucose. Hyperglycemia is to be defined in the study as greater than or equal to 250. Hypoglycemia is to be defined in the study as less than or equal to 50. And dysglycemia is either hypoglycemia or hyperglycemia. Now let's go to clinical looking glass. We're first going to build our medication sets. We need to get a medication set for Gatifloxacin and a medication set for the macrolides. So normally we go to the three-dotted button once we are building our cohorts, but I will show you another way to do this. You can go to Tools, Set Builder, left-click on Set Builder, and a window will open up, which is a Set Builder window, and now you have to specify what the set type is. Normally, when you use a three-dotted button, the three-dotted button knows where you're coming from. It knows what set you're going to need. In this case, we're going to have to define the set, and it's going to be a medication set. Now, medication sets are used both for the prescription event, which is the outpatient medication, for the medication event, which is the inpatient medication order, and for the administration internal medicine event. We're not going to choose an existing set. We're going to first look for Gatifloxacin. And this is going to look for the exact letters. You could put Gata, Gatiflox, let's find. So you type in the search option, Gatiflox, and find. Now what appears are these two Gatifloxacin. You can see one is clearly an intravenous medication, Gatifloxacin D5W. I'm going to include it as well as the usual Gatifloxacin because when I use the prescription order, it will basically ignore the D5W if it hasn't been prescribed in the outpatient. So I could highlight each one with a control hold and then use add, or I just hit add all. And now I'm going to save it. I'm going to call this Gatifloxacin. Now, for macrolide, let's clear it. For the macrolide set, maybe you'd be tempted to type macrolide and find it, but you wouldn't find any macrolide. This is a class of drug. You'd have to know there's erythromycin, there's azithromycin, there is clarithromycin. It's all part of the set. If, however, you don't know what are the different macrolide drugs that you want, you have to basically explore the data set. We use FDB in our system here, so we can explore. You explore at different levels of aggregation. So first we take a look at the first level, and we're told there's something called a therapeutic class. We hit go. Now all the medications that fit into a therapeutic class are here. This is a huge set of medications. You can choose alternative therapies, anti-infective agents, anti-neoplastics. Clearly we're dealing with antimicrobials, so it'll be an anti-infective agent, and we'll hit go. Now it shows up in the Explorer list are a whole bunch of anti-infectives. But if you take a look within that group, you have antibacterials, antifungals. We're going to choose antibacterials. Go. And now within the antibacterial at that level, we have a whole bunch of antibacterials. And here we have something called macrolide antibiotics and combinations. Go. Now it's showing up are just the macrolide antibiotics and their combinations. If we want only the macrolides, I click on that and I hit go, and now I have a set of just the macrolides. Azithro, clarithro, erythro, and these other agents. I'm going to add all of them. In fact, I'm going to just leave it with the erythromycin, the azithro, clarithro, and erythromycin. Get rid of these last two. I'll move this one as well. So basically I have azithromycin, clarithro, and erythromycin as my macrolides. I'm going to save it, and I'm going to save it as a macrolide. So now I've created my two sets, my macrolide set and my gatifloxacin set. And now I'm ready to actually go to my cohort. So let me now go to the event canvas. And I want to create a folder for my dysglycemic problem. I'm going to call my folder dysglycemia. And now I'm going to build my cohorts within it. Cohort. First my gatifloxacin. And I want to call this gatifloxacin. I want my earliest time they were prescribed this drug. U event definition. U event definition is gatifloxacin outpatient prescription. I choose from prescription start. My definition is I'm adding a condition. My condition is going to be med order type. Normally I would hit the 3 button and build from my set builder right here, but I already have, as you can see, the gatifloxacin and macrolide that I built before. Update and close. Update and close. My duration definition. January 04 to March 1. included because it has a left bracket. Update and close. Duration is now populated. Now we're going to do demographics. The new demographic is going to be age greater than 65. Update and close. So we have the three pieces: prescription Gatiflox between January 4th to March 6th, age greater than 65, and it's the earliest. Update and close. Here's earliest of any, and is with good practice, I'm always making it point to the line I'm interested in. I'm ready to build. 2,551 patients. Now I want to build my macrolid, and I want to do this quickly. So I rename it macrolid. I choose to save as so it won't overwrite. And now I'm only going to edit this line, and I'm going to modify the event definition. Event definition is going to be macrolid. I'm going to, of course, it's going to be prescription again. Medication order I'm going to edit. Edit is now going to be a set called macrolid. I'm going to left update and close. Left click update and close. Rename this macrolid. You can see earliest macrolid January 04 to March 606, age greater than 65. Update and close. And we're going to build it. 2,271 patients. So we've now built both our cohorts of patients, those on macrolids, those on Gatifloxacin. Now we have to build our study. We're going to go to Study Designer. I'm going to add a new study. I'm going to name the study Gatifloxacin Dysglycemia. I'm going to have to enroll my two cohorts. My baseline is going to be the macrolid group. Because we're going to be comparing the hyperglycemia against the macrolid baseline. I'm going to add another group. That group is going to be my Gatifloxacin group. My method. It's going to be a time to outcome. The time to outcome is going to be hyperglycemia. And here's our racetrack paradigm. We don't want to start on day one when the person was prescribed the medication because he hasn't had time yet to pick up the medication or take it for any length of time. So we want to activate a blackout period. That is, we want to follow each patient after the first 2 days. Therefore, blackout period is 2 days, 0 to 2 days. I want to get the time to the first hyperglycemic event so I can use the simple method for this. I'm going to look for a 30-day follow-up window. The outcome is hyperglycemia. I don't have this as an option. I'm going to build it now. Hyperglycemia. This is going to be lab test. Add condition. Lab test type equals to, it's glucose combo, so I'm going to put gluc and then go down, look for it. It's glucose combo, FSG plus blood. I'm going to add another criteria, and that additional criteria is I want the glucose combo to have a value. That value has to be greater than or equal to 250. Update and close. That's my event definition. Save it. It appears here as hyperglycemia. OK. I exit. Now I find in my drop-down hyperglycemia. Hyperglycemia simple. I run the method. So you have your breakdown, 703 macrolid males, 1568 macrolid females, 926 Gatiflox males, 1624 Gatiflox females. Target event. We notice the two lines separate. The p-value is very significant, less than 0, and we see the Gatiflox goes on to hyperglycemia much more quickly. In fact, at 30 days, the macrolid is 2.6% have an episode of hyperglycemia. The Gatiflox has 9.7%. The confidence interval of the macrolid at the upper level is 3.3, whereas the Gatiflox at the lower level is 8.1. It doesn't overlap. Now, once again, we notice all these empty values, 60, 90, 180, and 365 days. That's due to the fact that we have the wrong interval in our criteria. Let's go back to our criteria. Notice when we built it, we said estimation points were 30, 60, 80, 180, 365. We really should have said, let's have the estimate at 3 days, 5 days, 7 days, 15 days, 20 days, 25 days, and 30 days. That's really the interval we're really interested in. Now let's run it again. Take a look at the target events, and now you see all the values filled in. You see consistently Gatifloxacin has a higher attack rate of hyperglycemia, highly significant. All events really doesn't tell us very much. It tells us how many measurements of high blood sugar there were, but sometimes you measure more frequently because it's higher, so I'm not sure it helps you very much. But the target event is the cumulative percent who've had at least one elevated glucose. And you see that by day 30, the macrolid group only has 2.6% of their population with elevated glucose. The Gatifloxacin group has 9.2%. If you take a look at the demographics, we see the breakdown has more males than the Gatifloxacin. And we also see that the age is a little older in the Gatifloxacin group. So there might be additional things you want to adjust for in modeling, but it's a useful first cut of this problem. As part of this exercise, I ask you to do an advanced method on this. Repeat this with the advanced method. So let's do that together. We're going to have a time to outcome again. The method is going to be hyperglycemia advanced. We're going to go to the advanced paradigm. Again, we're going to ask for the blackout period of 2 days. Now, however, we have to actually build our analysis definition because we don't have one yet. We're opening up the analysis definition block. We see it's not properly centered. We double click on that, double click again. So now we're ready to build. Hyperglycemia, first 0 to 30 days after. Is it within condition? I hyperglycemia, earliest, new event definition, now we have to define it. Lab test type equals to, start with GLUC, GLUC, then just flip down. Adjust flip down, there's glucose combination, finger stick and blood glucose. Now we have to add another condition. That additional condition is lab test value greater than or equal to 250. Left click on update and close. Left click on update and close. We want this to be this to be 0 to 30 days after the cohort collection. That's the period of time we're going to follow. Remember, the blackout period is being controlled by that left flag. Update and close. We have to make the first line also edited. We need to edit the first line and move it from all to earliest. It will not show up in a time to outcome if it has all in there. And point to hyperglycemia. Update and close. And save it. And it appears here. Now we're going back to the study designer. You see in the drop down, should be the last one there, it is. Put in our table, 3, 5, 7, 10, 15, 20, 25, and 30. Call it hyperglycemia. Advanced. Now we're ready to run. Once again, we see how this is significant. We see the 2.6% to the 9.2%. We're going to go between the two pictures. Just flip back and forth, and you see the images are exactly the same. So with either the simple method or the advanced method, it looks exactly the same. You've seen the first 0 to 2 days, then no events at all, because it's a blackout period, so it's always going to be flat. Now we want to consider dysglycemia, and dysglycemia is either hyper or hypoglycemia. Let's use a simple method for that. Time to outcome, dysglycemia simple. Adding another method. Blackout period again is 2 days. I'm going to follow people for 30 days. Now we need to create our dysglycemic event. But we already have hyperglycemia. But that really doesn't help us. So let's see how we would have to build a dysglycemia. First step. It's going to be a lab test. This is what both of them have in common. The lab test condition is lab test type equals to type glucose and so finger stick glucose or blood glucose. And now we have two conditions, right? You either can have hyperglycemia, that's greater than or equal to 250, or you can have dysglycemia, which is less than or equal to 50. How do you use both conditions? It's really sort of a parenthetical. I want the type equals to glucose and, parentheses, glucose greater than or equal to 250, or glucose less than or equal to 50. So in order to create that parenthetical, you need a subgroup. We hover over the definition, we right-click, and now you see the add condition. We're going to add a subgroup. And the subgroup, we're going to give it a name. Subgroup's name is going to be dysglycemia. Or better yet, we'll say hypo or hyperglycemia. Okay? And we want an or condition behind it, because you're either hypo or hyperglycemia. This or condition is controlling what the different condition lines within the group will be. Update and close. Here's the or condition. Now we hover over the index line for the group. Notice the parentheses indicating this is like a parenthetical. Hover over the group. Notice how a box appears around it. Right-click on it, and we're going to add a condition. And the condition is going to be a lab test value. Lab test value is less than or equal to 50. Right? That's a hypoglycemia. Now we're going to hover over that index line again where the parenthesis is, right-click there, add another condition line, and that condition line is going to be lab test value greater than or equal to 250. That's a hyperglycemia. So you have the first line, which is shared by both. Both of them are, the first line is glucose, either fingerstick glucose or blood glucose. And the other criteria is built within a parentheses or a subgroup, lab test value less than or equal to 50, or lab test value greater than or equal to 250. And that's dysglycemia. Look over here. You don't see it yet in the management pane. We're going to save and exit. Definition saved. And there's our dysglycemia. Let's exit this. Let's choose from the drop-down dysglycemia. We're ready to go. Let's just change the days again to be 3, 5, 7, 10, 15, 25, and 30. And let's run it. Now what you would expect, since you have outcomes that are either hyperglycemia or hypoglycemia, you would expect additional results, more people. Let's see what happens. So in fact, at 30 days, we can see again the gatifloxacin group is achieving the target faster than the macrolide group, highly statistically significant. We see 3% of the macrolide group and 9.9% of the gatifloxacin group hit either hyper or hypoglycemia. And compared to what we had before, it was 2.4 and 9.2. 2.4, 9.2, 3.0, 9.9. So you get a little bit of a bump from those who are hypoglycemic. Let's save this. Now we can't completely ignore the need to control for some confounders. Right? We have the age differential in the two groups, and we're not going to try to do a huge statistical analysis here. But what we will do is we're at least going to look to make sure that there were not a maldistribution of diabetics in the two populations. That is, you wouldn't want to have one group overrepresented by diabetics than the other. So in order to do that, we actually want to take a look and see how many patients were diabetics in the two groups. One way to do this is to make a list method, which is what we're going to do now. We're going to make a list method, and we're going to look for the presence of hemoglobin A1C greater than or equal to 6.5, which is the definition of diabetes according to the ADA. So we could do this in the simple method. I'm going to do it in the advanced method just so that you get more exercise this way. Open up the triple button, double click here, double click again. Hemoglobin A1C greater than or equal to 6.5, 730 days before. I want to find those people of hemoglobin A1Cs greater than or equal to 6.5, 730 days before. So I want to edit this. I want to make this the earliest. Well, it really doesn't matter. I only want to know whether there was anyone at all. So I'm going to say the latest. Update and close. I'm going to hover over it. I'm going to add a condition line within. And the within condition line is going to have hemoglobin A1C greater than or equal to 6.5. Call this latest. New event definition. The new event definition is going to be a hemoglobin A1C greater than or equal to 6.5. Lab test. Add condition. Lab test type equal to hemoglobin A1C. Now I add, right click on the definition, left click on add condition. The condition is going to be lab test value greater than or equal to 6.5. Update and close. Update and close. We want it 0 to 730 days before the cohort collection date. And I'm going to save that. Look over here, see if it shows up. Oh, actually, before I save it, let me change my first line, just as good practice. Latest of event hemoglobin A1C. Update and close. Now I'm going to save it. And we see it here. Hemoglobin A1C greater than or equal to 6.5, 730 days before. This is the latest one. We'll choose it from the drop down. Notice how the metaphor shows you that you're going back in time. This is a list method. As you recall, we have the list populator. We're going to maximize the list populator. It is a singularity. We're looking only for one hemoglobin A1C, so we're going to use the wide view. I want to get rid of almost all these other things just to make the point of what we're looking for. I'm getting rid of all the other ones, so I'm only going to have lab test type, lab test value. I'm also going to have something called count that I can bring over. Count is a very nice thing. It counts the number of positives that are there. So in this case, in singularity, 1 would mean there's a value, 0 would mean there's no value. That is, we have evidence of a hemoglobin A1C, that's a 1. 0 means there's no evidence of a hemoglobin A1C there. It's good for a logistic model. You have a quick way to be able to cross-tab on the count variable. So you have the actual lab test value and the count. And now we're ready to run the method. Now I could also get in my spreadsheet the language, the SES score, all sorts of other variables, but for the purpose of this exercise I want to do this very quickly. I want to know whether there's an obvious difference in diabetes diagnoses in these two groups. And here what you see is you see the count variable. 1 means there was a hemoglobin A1C. Here's a person who had no hemoglobin A1C that met the criteria. And here's the actual value, 7.1, 6.6. This one never had a hemoglobin A1C that met the criteria. So we have some that have and some that don't. So in one spreadsheet, you have all that information. Now we want to export it. Excel 2007, export, data file. Open. We have the summary for group 1, the macrolide, summary for group 2, the gatifloxacin, summary for what was the criteria for the AD, in this case, hemoglobin A1C greater than or equal to 6.5. And we have the raw data. I'm going to rename this count so it'll be easy to work with. Lab test, I'm going to rename this. Hemoglobin A1C value. And now that I've renamed the titles, I'm now ready to use some of the powerful features of Excel. I'd suggest you learn them. I'm going to insert, it's called a pivot table. Left click on pivot table. It automatically looks at the entire spreadsheet. I hit OK. And now it lets me actually look to build a pivot table. I can bring group numbers into rows. Here's group 1 and group 2. That's the macrolide. That's 2 is the gatiflox. I bring group number into values, and it's counting. There are 2,271 people here and 2,551 here. And if I want to have the count, whether there was a hemoglobin A1C or not, I can move that to the column. And here you have the count of people who had no hemoglobin A1C of that value, and the count that did have it of that value. This gives you the absolute number. You can click into a cell, left click in a cell, right click in it, and then you can hover over show values as % of row. So now you see that 22% of the macrolides had a hemoglobin A1C greater than or equal to 6.5, and 22.78% of the gatifloxacins had a hemoglobin A1C greater than 6.5 in the 2-year period before. So we don't have any evidence of a dramatic difference in diabetes diagnoses through the criteria of hemoglobin A1C, and at least that confounder is addressed to some extent. So now you've had an opportunity to recapitulate a study in the medical literature using data from Montefiore itself. You have been able to identify the effect of gatifloxacin on hyper or hypoglycemia in the elderly population, and you have controlled for a confounder.